All right, so let's go ahead and skip over to this slide here. This slide is taken from your book, Figure 218 on page 33. And what it represents is our sensitivity to different wavelengths of light as a function of the wavelength, right? So for the top graph, right, we have going across the x-axis, we have wavelength, right? Remember humans can see from about 400 nanometers to 700 nanometers, and these are the colors that are represented by each one of those different wavelengths. So the shorter wavelengths are going to look bluish, and the longer wavelengths are going to look red. That's what it's going to appear like. And then we have threshold. In other words, the level at which you can detect the stimulus. And as you can see, our threshold for colored light, for visible light, is highest at around in the upper 500s. So in the upper 500s, that is where we are the most sensitive uh, to light. So we see best the yellows and the oranges and the greens. Okay, those are the colors that we see the best, which makes sense, right? Because these are colors that are found in nature, right? Color of plants, the color of fruits and vegetables, the color of dangerous things like dangerous plants and dangerous animals, all have colors right around this area. Okay, so it makes sense that that would be our most sensitive the wavelengths that would have the lowest thresholds to be activated. Similarly, we also have a spectral sensitivity curve, which spectra we are most sensitive to, and it of course mirrors perfectly the threshold curve, right? So we don't see the blues so good. We don't see the deep reds so good, but we are highly sensitive again to these colors in the middle of the visual spectrum. Yellowish, uh, the yellowish colors, the yellowish greenish colors, and the yellowish orangish colors. Those are the ones that we are the most sensitive to. Okay. Um, Rods are much more sensitive to short wavelength light. In other words, light that is on the bluer side of things, right around 500 nanometers, so right around here. That's where rods see best, right? Cones are more sensitive, a little bit higher at 560 nanometers, right? There's this really cool effect called a Purkinje Shift, where you can enhance the sensitivity to the short wavelengths during dark adaptation. In other words, as your eyes become adapted to the dark, you get more sensitive to the around 500 nanometers instead of 560 nanometers, and you can do this. Uh, you can do this as a little experiment on yourself. So get yourself an eye patch and wear an eye patch for like 20 or 30 minutes. Right, just have it on your eye, and that'll make one of your eyes dark adapt. Right, so I do that, and then take the eye patch off, and then go and close one eye and open the other eye, and vice versa, and see how different things look. You're gonna see if you do that, you're gonna see that the stuff that you see out of the darkened eye, the one that you had the eye patch on, is going to be more. The colors are going to be more in the blues, more bluesy colors as opposed to the brighter colors, right? If you look, for example, at the picture on page 220, on page 34, figure 220, I think it's this picture right here. Uh, the bluer color, the flower that's on the right hand side, is going to look brighter to you with the darkened eye as opposed to the red flower, right? Because the blues are going to stand out more, and that is the Purkinje shift. Okay. Here's a picture, a diagram of the sensitivity, a relative sensitivity of rods versus cones, of course, right? And rods are more sensitive to those dark green, bluish colors, whereas cones, of course, as we said, are more sensitive to the yellowish, greenish colors there, right? There's the Purkinje Shift flowers, right? Spectral sensitivity. So what accounts for this different sensitivity, right? Why do we have cells in our retina that are sensitive to different wavelengths of light? Well, it's due to the pigments, the opsin and the retinol pigments that live in the cells in the rods and the cones. The pigments in the rods absorb photons that are traveling at 500 nanometers, at a wavelength of 500 nanometers. And the cone pigments absorb the photons that are best at 419, 531, and 558. Okay? Remember, cones are specialized for color vision. Rods only for dark vision, right? So it makes sense that rods really can only absorb at one color, and cones have different colors that they absorb at based on. Let's see, I forgot to set my clock. Sorry about that. Based on what color they're specialized for, right? Some cones are at 419, which is way on the low end, the blues area. Some are 531, which is in the middle, the yellowish area, and some are at 558, which is the reddish area. Okay, they work together to make up the colors from these three basic colors. Okay. All the cones, if you look at all the cones together, they're right around 560 nanometers. That yellowish, greenish area there on the curve, right? So here we have all of the different rods and cones, right? The sensitivities, right, of all the visual pigments, right? This is from your book on, again, page 34, figure 2.21, right? The rod pigment is here, right? That's where the rods are most sensitive, right? And then we have the three cone pigments, S, M, and L, medium, short, medium, and long wavelength. Okay. Short wavelength pigments are sensitive to the blues, right? The dark blues. The medium wavelengths are more sensitive to the greens, and the long wavelengths are more sensitive to more on the yellow reddish area. Okay. So remember, you have the rods are sensitive here, right? That's the proteins are most sensitive to this color of light at 500, and then your cones have three different sensitivities. Each cone is one of these. It's either a short wavelength cone, a medium wavelength cone, or a long wavelength cone. So your rods and cones pick up the light. They detect the light using this chemical reaction involving the retinol and the opsin that we've described. Now you have to remember, rods and cones are neurons. They're highly specialized neurons. Okay, so since they're neurons, they generate electrical signals and signal other neurons. They communicate with other neurons. That's what neurons do. That's their job. Okay. The major parts of a neuron that you should have learned in Psych 10 in 301, right, in introductory psychology. You got your cell body, your dendrites, and your axon, or your nerve fiber, right? So the cell body's usually pictured in the middle, right, and that's where all of the biochemical life-sustaining apparatus lives, including the nucleus and the DNA is in the cell body. Then you have the dendrites projecting out of the cell body, and typically the dendrites are in all neurons. The dendrites are designed for receiving signals. In the case of rods and cones, the dendrites have specialized to become the tubes and the little cone part that contains the proteins, the opsins and the retinols to look for light, right? That's what the dendrites do. And then the axon is the part of the neuron that projects away from the neuron and sends the signals to other neurons, okay? Sensory receptors are simply specialized neurons, like the rods and the cones, that are designed to absorb some kind of energy from the outside world. In the case of rods and cones, they're designed to absorb light energy and respond to it, right? So here's another example of a neuron. This neuron, this is actually two neurons. One is a touch receptor. Looks kind of like an onion or a grape, and we're going to talk about why it looks that way when we get to touch, right? This is a receptor that is specially designed to look for to feel pressure on your skin, okay? But once it gets that stimulus from the environment, it causes changes in the shapes here of this part of the receptor, which causes proteins to change and ion gates to open, and that causes a nerve to fire, and or that causes this receptor to fire an action potential down the length of its axon till it synapses here with another neuron that lives in your spinal cord. And this is a neuron that relays the information from the this is called usually referred to as an interneuron, right? It has dendrites that receive information from other neurons. In this case, it's receiving the information from this touch receptor, and it processes the information here in the cell body. And if it decides to fire off a signal, it does so, and it fires it down the axon. And that signal hops down the length of the axon until it gets to the dendrites and it synapses with other neurons. Once the information is once the light information is transduced into an electrical signal, it gets relayed into the brain, right? And it makes a lot of different stops in the brain, right? It travels along the optic nerve, and one of the places that the visual information goes is an area called the lateral geniculate nucleus, the LGN, right? This is one of many places that the information goes, but the thalamus is a large structure in your brain that seems to be a relay point for sensory information. So all the sensory information that you get from your body, whether it's sight, sound, hearing, touch, smell, most all of it goes at some point into the lateral geniculate nucleus to be processed. From there, it gets relayed to the visual receiving area, the visual cortex, right there in the back of your head, right here in the occipital lobe of your brain, of your cortex. And then from there, that information it's processed, analyzed, and then from there it gets passed on to more forward areas of your brain for further analysis and to make decisions as to what you're going to do with it. Neurons are really, really small. They're microscopic, right? You have to look at them under very powerful microscopes to be able to see them. However, just because they're small doesn't mean we can't record a signal from a single individual neuron, and we can do that. We've had that technology for a very long time. For about 50 years, we've been able to record signals inside of different neurons. And basically, what they do is they insert a tiny thin glass electrode inside a nerve fiber, and then they have another electrode outside the nerve, right? So if this is the nerve fiber, right? Imagine it as a tube, right? Let me see if I have a tube here I can use. Well, here's one. Okay, so here's a tube, right? So here's a tube, right? So here's a nerve fiber. And so what they do is they insert an electrode inside the nerve fiber, and then they have another electrode that's sitting outside the nerve fiber. So there's one in the nerve that's stuck in the nerve fiber, and there's one outside. Well, if you know anything about electricity, that's how you measure electrical potentials, is the difference between two electrodes, right? And we can actually measure the electrical charge difference between the inside and the outside of that neuron. And once you measure it, it's about 70 millivolts. There's a negative 70 millivolt difference between the inside and the outside. So the inside of the neuron is negatively charged, and the outside of the neuron is more positive, right? And the difference, if you measure it in volts, it's about minus 70 millivolts. So it's like a little battery. It's like a little capacitor that holds a charge, right? And that charge is about minus 70 millivolts.